Welcome to my talk. My name is Susanne Renner. I'm currently a research scholar at Washington University in St. Louis. Between 2003 and late last year, I was professor of systematic botany and mycology at the University of Munich and director of the Munich Herbarium and Botanical Garden. I met two of the organizers of this workshop, or this is the second workshop of these two workshops, namely Rudy Ammann and Ramon Rossello Mora, in 2012 at three workshops in Germany on challenges for taxonomic research in the age of omics technologies. And these workshops were so interesting and so inspiring that I then sat down and wrote a grant proposal which was funded. And so I was speaker for this program that ran between 2017 and 2020 with the title Taxonomics, New Approaches for Discovering and Naming Biodiversity. And what was special about this program was that for the first time, the DFG funded a thing called focused on systematists, and that included zoologists, botanists, and mycologists. This was really our selling point, and of course we were held together by genomes. One of the outcomes from this project was this paper about the position of taxonomic data such that they will be useful in the future. It's really focused on databases and how they at the current time do not fit well with many kinds of taxonomic data, for example, images. And in our paper, we thank Christopher Lane, who is here today, and I really look forward to meeting him. So Linnaeus named 13,400 species of animals, plants, and fungi. How did he do it? He did it by doing it. So the way he did it was that he used specimens and images were available. He did not like descriptions. Instead, he used diagnoses of 2 to maximally 12 words. And all of this approach that he used is described in Philosophia Botanica and also other papers because he was very proud of the approach. So here's an example of one of the species that Linnaeus named, namely Volvox globator, which he studied under his microscope. You see here on the right Linnaeus's microscope, and then we can read the text he wrote about this animal, or this algae that he thought was an animal. And I'm translating. It has free gelatinous round body without limbs, and within it are swimming a few round offspring. And he cites a drawing by Henry Baker and another drawing by Russell Frun. In most cases, however, he preferred just a few words, as I said before. For example, here's his diagnosis of the cucumber, Cucumis sativus. It has narrow leaves, straight leaves, and oblong fruits. And that's it. And then he has again two illustrations, one by Bohin and some others cited here above. And he didn't know the habitat because it was a cultivated. And because this was Linnaeus's approach and he named 13,400 species, it is very important for the codes, or at least most codes, to be clear about what a diagnosis is. For example, our code, the one for algae, fungi, and plants, states a diagnosis of a taxon is a statement of that which, in the opinion of its author, distinguishes the taxon from others, as we just saw in the case of the cucumber. In the zoological code, this is phrased as follows. When describing a new taxon, an author should make clear his or her purpose to differentiate the taxon by including with it a diagnosis, that is to say, a summary of the characters that differentiate the new nominal taxon from related taxa. In a description, of course, you list all sorts of stuff that is the same in all the taxa in a group. Let's say they all have leaves or opposite leaves, whereas a diagnosis is the specific thing that sets the new taxon apart from the already known ones that are similar. And because this is Linnaeus's approach, all the codes say that a description is voluntary. Here you have the code, a screenshot from the code for plants, where it says Latin description or diagnosis. This was the case until the end of 2012. Now it could be in English description or diagnosis. And the same in the zoological code. You can either write a description or do a short definition. And this is also the case in the new fungal code, which was just developed in 2018-2019. Again, description or a diagnosis, and the diagnosis could be 2 words. So the codes for animals, plants, and fungi all need to allow illustrations as nomenclatural types, because in the case, as you saw in the case of Volvox, Linnaeus used illustrations wherever he could, and diagnosis of a few words as protologs, so as not to lose Linnaeus's 13,400 names. Now, the current code for prokaryotes does not mention diagnosis. You only have description, which is, in my opinion, an unnecessary difference. Now, Linnaeus's short, sharp diagnosis, of course, bring to mind DNA, traits and genomes or traits and genes that provide clear, sharp diagnoses, setting related things apart from each other. And so I reviewed this topic in 2016, now 4 years ago, and by that time I found that between 2001 and 2015, 75 new species had been formally named with DNA-based diagnosis. And on my next 5 slides, I will show examples. These species come from the worms, the flowering plants, other worms, annelids, spiders, arthropods, chordates, and the example I will give comes from the turtles, butterflies, lichens, mollusks, nudibranchia. I'm going to show a picture, and by that time also a pathogenic fungus, and the number of species with molecular diagnosis was increasing very fast. So here's my first example. Here's this New Zealand centipede, and the diagnosis says that it has an adenine in position so-and-so after primer so-and-so in 18S ribosomal RNA, and there are 3 thymines in position blah blah blah, and it also has dorsal and ventral supernumerary malphidian tubules, both oriented anteriorly. Now here's my second example. There's a bunch of snails, marine snails with reduced and variable morphological characters, but differentiated by a unique combination of nucleotides and cytochrome B, etc. Here's my third example. It is this wonderful nudibranch, and the diagnosis says that this species can be distinguished by 70 molecular diagnostic characters in cytochrome oxidase 1 and 39 in the 16S ribosomal RNA, and morphologically it is characterized by this and that. And here's my fourth example. These are African turtles, and they have various morphological characters, but also molecular characters like a gap instead of an adenine cytosine and thymine in position so-and-so and whatnot and whatnot. Now, DNA-based diagnosis, of which I have now given you 5 examples, but there are about 150 of these by now, are not the same as allowing genomes as the only type material, but they touch on a related issue, as will become clear in a minute. The Code of Zoological Nomenclature already permits DNA as sole type material in certain positions that are spelled out in their Declaration 45, which I will show in my next slide. Now, this is a page from the current Code of Zoological Nomenclature, which is online, and you can just search for DNA type or Declaration 45, and it sets out here in these recommendations under which circumstances DNA as the sole type is permitted. And because we don't have time to read all this stuff, I will paraphrase the gist of this on my next slide. So what these recommendations say is explained here by Thomas Pape, the president of the Commission for the International Code of Zoological Nomenclature, and he emailed me this explanation. The zoological code still requires that a new species has a physical name-bearing type, but this type could in principle have disappeared, disintegrated, liberated into nature or something similar. And I want to stop here a second and move your eyes or ask you to look at these images of these mammals. These are new species of mammals that were described, named in the past 10 years. And of course, these are all rare, tropical, many times nocturnal animals. And we can understand their behavior sometimes from cameras that have, from traps, from genomes recovered and assembled from their poop. We know that these are new species. The people who study these animals know that these are new species, but they would sure not kill one of these animals to make a specimen. It would be completely unethical. You might capture it and get a sample of hair or tissue or something. Okay, so this would be an example of the animal being liberated into nature. Okay, and now let me read on. The recent fire at the Museo Nacional in Rio did not invalidate those new species in press or in prep for which the holotypes went up in flames. This also means that the holotype that was entirely destroyed during DNA extraction, and examples of this exist, they are listed in my earlier review there of DNA diagnoses, would still count as the name-bearing type, although the only evidence of that specimen would be nucleotide sequences. Therefore, although the sequences by themselves cannot be types, they would be de facto types. Now, I am convinced that DNA sequences, best of all, of course, entirely assembled nuclear genomes, but we don't have that many yet for plants, should be permitted to serve as nomenclatural types. And I have therefore submitted a proposal, a formal proposal, to permit DNA sequences as types when preservation of specimens is not feasible. I submitted this to the rapporteurs, Nick Torland and Ugo Versema, who are working on the next edition of the Code for Plants and Algae. What I'm showing you here is not quite final, although I did submit it. I just want to read the 4 lines in red from my proposal. I agree with these authors, being Torland and Versema, that DNA sequence is analogous to an illustration in that it depicts the features of an organism. In addition, it is readily preserved and shared 24/7, 365, and thereby useful as we strive to name the Earth's biodiversity. As argued before, it would not be a great change in the code to allow nuclear DNA sequences as nomenclatural type under restricted circumstances. And then I have some recommendations that are here in tiny print that are basically or almost copied from the recommendations of the zoologists, because I think it would be great to bring the International Code of Nomenclature for Algae, Fungi, and Plants in line with the International Code of Zoological Nomenclature. So to conclude, language is of course metaphysics and serves communication, as others in the first workshop have already stressed. Nomenclature provides the rules for linking metaphysics to physics, and the type method of attaching names to a specific stored and retrievable onomatophore, this is the technical term for a name-bearing thing, is the best thing we have, and all 5 or 6 current codes could actually allow this easily. Thank you.